Good morning, everyone. I hope that everyone can hear me fine. Yes, good, thank you. So my name is Elena Comelli. I'm an associate professor in the Department of Nutritional Sciences at the University of Toronto and at the Lawson Center for Child Nutrition and Health. I'm delighted to welcome you to the fourth Lawson Microbiome Nutrition Seminar. This year, our seminar is special. This is our first virtual seminar, and this also allowed us to host two international speakers, Dr. Collado and Dr. Selma Royo. We are so happy that you were able to accept our invitation. We also have a very diversified audience this year with attendees from multiple universities here in Canada and abroad, and we are very excited about this. We are looking forward to a great discussion at the end of the talks. Before we start, I would like to thank Joanna and Brian Lawson for making this possible. I know that we tell you every year, Joanna and Brian, but it is every year that we feel grateful for your vision and your support to microbiome and nutrition science. So for this, we thank you. I have, nice to see you. We have a few housekeeping notes before we start. So the webinar is being recorded and it will be posted to the Lawson Center website. And I would also like to ask you to please ensure that your video and microphone are turned off during the presentations. We will have a Q&A session at the end of the presentations together. So the two speakers will speak in a row and then we'll have a discussion at the end as a panel. I invite you to put your questions in the chat, but we would be happy if in the end you would want to ask your questions yourselves. And I will moderate the session, but to have some discussion and more personal interaction, even in this setting, we'd be happy if you wanted to ask your questions yourselves. Okay, so I would move now to introduce our first speaker, Dr. Marta Selma Royo. Dr. Selma Royo holds a PhD in biotechnology, and she's currently a postdoctoral researcher at the Department of Biotechnology, Institute of Agrochemistry and Food Technology at the Spanish National Research Council in Valencia, in Spain. The work of Dr. Selma Royo focuses around the effects of perinatal factors on microbial colonization and on maternal and infant health, and she implements multidisciplinary approaches, including computational analysis, microbiology, and cell culture model. Dr. Selma Royo, we are looking forward to your talk.
Can you hear me?
Yes, we can.
Thank you. Thank you for the presentation. And also thank you for the invitation to present part of the work of my PhD in the Mari Carmen Collado's lab. So I'm going to share. Yeah. Okay. So today I'm presenting, as I said, the work that I perform in the Mari Carmen's lab during my PhD, and it's focused on the importance of the perinatal environment for the microbial colonization and cell development. So human being has defined it as a large brain bipedal primate with the capacity for language and use of complex tools. However, the advances in sequencing technology and also the results from the big projects in microbiome has suggested that the definition could be incomplete and we need to add a few words more. And human beings consist also in the community of microbes that inhabit its body and contribute to the maintenance of the host homeostasis. It has been estimated that around 20% of the microbiota it's in the mucosal regions, being the gastrointestinal tract the most important niche in the human body. And there, microbiota participates in several functions, including metabolic, structural, and protective processes. So due to the importance of microbiota for human health, several observational studies have been performed, and the results reported a link between alterations in microbiota during early life with some diseases in adulthood, such as allergy or obesity. This observational study has triggered the inclusion of microbiota as part of the developmental origins of health and disease hypothesis. And this inclusion is based on the idea that during early life, the recognition of not only what to attack, but also what to tolerate would be essential for the correct establishment of the symbiotic relationship between host and microbes. It's important to keep in mind that colonization runs in parallel with immune system modulation, and animal studies have highlighted the essential role of microbes shaping the immune system modulation. So we can think about it, the microbial colonization as a war or invasion, but the results of this study has suggested that it would be more like a stepwise process in which every every participant is interconnected, and every step has its time and its process. So despite of the fact that the microbiota is changing through life in childhood, the microbiota is extremely variable and sensitive to external factors. And for that reason, the infancy has been considered a window of opportunity to influence the infant development, adult health, through a modulation or alteration of colonization process. So as I mentioned, the colonization is an extremely complex process, and it's influenced by several factors. The most studied are mode of delivery, gestational age, or infant feeding. And these factors are going to influence not only infant microbiota, but also infant development and immune system maturation. At this point, I wanted to highlight the importance to study also the maternal microbiota, since it is the first bacterial source of the infant colonization. And these factors, some of these factors are going to influence also the maternal microbial communities. So in this regard, several questions remain unanswered, including how the perinatal factors influence the maternal microbiota and which molecular mechanisms are involved in this effect in maternal microbiota, but also in the colonization process, and which molecular mechanisms are behind the observational studies that report this association between disease and alteration in the colonization process. So in this context, my work in the Mari Carmen's lab is focused on the first month of life, and the general aim of my PhD project was to determine the impact of these perinatal factors on both maternal and infant microbiota, as well as the health outcomes of the mother-infant pairs. For this purpose, we have a cohort with 250 modest infant pairs and samples collected at different time points. I think Mari Carmen is explaining the characteristic cohort in her talk later. So today I'm going to talk about the last work that I performed during my PhD, and is related with perinatal environment and the microbial colonization with infant growth. This is an overview of the methodology that we use in this project. We included 180 neonates in the analysis, born at hospital by both elective C-sections and vaginal delivery. In this study, we include also a subset of mothers that had a home birth. Home birth practices prevalence is increasing. However, the effect of this practice in colonization and also in infant health has been underexplored. So we decided to include this group of neonates in the study. And we collected also the clinical records of the participants from birth to 18 months of life. And we performed the 16S RNA gene sequencing, and we confirmed some of the results obtained by this technique with the quantitative PCR. Here I'm shown it's shown the flow chart of the study cohort. We have some of the neonates that complete all the follow-up of the study. Some of the neonates were recruited after delivery. So we have the samples from 7 days to 1 month of life. And some of the neonates participate only at hospital and decide not to continue with the study. And this is probably one of the main problems in the observational and longitudinal studies. And for sure, some of you experience this problem, the drop off of the participants in the big cohort. So regarding the results, at birth, we saw that the main contributor for the infant microbial composition was the place of delivery, showing the children that born at hospital higher richness and also higher relative abundance of immunity compared to those born at home. At the following time points, we found that the mode of delivery was the main contributor to the infant microbiota, and here we can see the evolution of the colonization at phylum level. And we observed that vaginal delivered infants at hospital shown an intermediate pattern somewhere between those born at C-section and those born at home birth. Those children show higher actinobacterial relative abundance, mainly due to the presence of higher bifidobacterium, and this result was also confirmed by qPCR analysis. Vaginal delivered infants, both home birth and at hospital, show lower relative abundance of Firmicutes and Proteobacteria than those that born by C-section. So the next question was, does it mean something? There is a biological meaning of these microbial differences in the colonization process, and we reported higher BMI zeta scores and also weight for length zeta scores from at 1 year and also 18 months of life of neonates born at hospital compared to those born at home after the adjustment of covariables. And it has, in this regard, it has been suggested that while high doses of antibiotics could be related with malnutrition and weight loss, the low doses of antibiotics could be related with weight gain and could be an hypothesis of the mechanisms behind the association between antibiotic intake and obesity later in life. So we continued exploring this hypothesis, and I'd be happy to hear your ideas or your comments about this. And then we wanted to analyze the possible mechanisms behind our results in microbial colonization, the difference according to groups. And for that purpose, we designed two kind of experiments, one of short-term exposures of 24 hours using fecal supernatant on intestinal and an intestinal cell line, and also an immune cell line, a macrophage-like cell line. And we measured the cytokine production and also the gene expression. And then we designed a long-term exposure, of 7 days experiment, and a triple co-culture model in a transwell system. In the basal compartment, we seeded the macrophage-like cells, and in the apical compartment, we seeded we tried to make an simulated epithelium system with an intestinal cell line and a globed cell line. We measure in this system the cytokine production, the gene expression, and also some functional assays such as barrier integrity or intestinal alkaline phosphatase production. And we use fecal supernatant at 1 month of from the neonates at 1 month of life samples, and we refreshed this fecal supernatant every day during 7 days of experiment. So in the intestinal cell line, in the short-term exposure of 24 hours, we found that all fecal supernatant induced the production of IL-8 and TNF-alpha with no differences between the groups. And at gene expression level, we reported an upregulation of the TLR4 root genes in those cells treated with home birth samples, something that was not observed in the cells treated with C-section samples. In the macrophage-like cell line, we found that this cell line was more responsive or more sensitive to the fecal supernatant than the intestinal cell line. And we found a higher immunostimulatory effect of the home birth samples compared to both vaginal delivered at hospital and C-section samples in terms of both IL-6 and IL-8 production. In the case of TNF-alpha, we saw a similar behavior of both samples from vaginal delivered infants. In terms of gene expression, we found a downregulation of TLR4 gene in those samples treated with C-section samples compared to the control. Sorry. So in the triple co-culture system, in the apical compartment, we measured the transepithelial electrical resistance as a measure of barrier integrity, and we found that the models treated with home birth samples show higher transepithelial electrical resistance compared to both vaginal delivered at hospital and C-sections. And this result was confirmed by the study of the transport of Lucifer yellow through the membrane, that showing the models treated with home birth samples show a transport of Lucifer yellow through the membrane. Regarding intestinal alkaline phosphatase, we observe a higher production of this marker of intestinal maturation in the apical compartment, showing the polarization of our simulated intestinal epithelium. And we found at the end of the treatment higher intestinal alkaline phosphatase activity in those models treated with home birth samples compared to both C-section and vaginal delivery infants at hospital. In the vessel compartment, we measured the cytokine release, and we did not find a difference according to studied groups in terms of cytokine production, probably showing the importance of the maturation of the membrane in the industrial barrier in early life to protect or to reduce the inflammatory signal in the neonate. However, we found differences in the trajectories of the cytokine release during the 7 days of treatment. So, as a summary of the results, we found higher Klebsiella and Clostridium genera in those children born by C-section compared to those born at home birth. I do not show the result, but we confirm by qPCR that those children show also lower bacterial load than those born by home birth at home. And those children born at home show higher relative abundance of bifidobacteria and bacteroidetes and higher bacterial load. And we hypothesize that this could be also related with an enhanced epithelial barrier function and intestinal maturation, as well as higher immune stimulation. Our results suggest a possible link and mechanisms behind the association between the C-section and the intervention during delivery and the first month of life with an altered intestinal and immune system maturation. At this point, I wanted to clarify two important aspects about the results. The first one would be that when we talk about home birth and C-section, we are not talking only about a place and a mode of delivery, and we could not separate the codominant variables such as the antibiotic use, the no antibiotic use in the home birth neonates, the home environment, probably a higher rate of breastfeeding since it has been reported that C-section born infants show a delayed initiation of breastfeeding, and also the higher maternal contact in those children born at home. And I have not put this in the slide, but also for sure the mothers that decide a home birth probably showed different lifestyle, different diets compared to those that maybe choose an elective C-section. And these factors need to be considered when these results are analyzed. And the second aspect that I want to mention is that we are not recommending a home birth instead of an hospital delivery, since we are focusing only in one aspect, the microbiome and the infant colonization, and several variables needs to be considered in this aspect, especially the maternal and infant health and security. So we are not recommending the home birth instead of an hospital birth. However, these results and others maybe could help to design strategies and policies in the hospital environment to favor the maternal-infant contact and bacterial transference and the possible consequences to avoid or diminishing this bacterial transference. So what are we doing now? What's next? As I mentioned, we consider especially relevant to study the maternal microbiota and how the prenatal factors influence it. So we are now on working on this. And also we are running experiments, further mechanisms studies with these samples to explore the molecular pathways in more complex models, cellular models, and also clarified the specific role of each bacterial and its taxa or its consortium of bacteria in the infant maturation. And for that purpose, we are starting with the metagenomic sequencing of the samples. And also we have in our lab a collection of isolates from those samples, and we are running now experiments to try to understand how each bacterial taxa could interact with the host. And now, finally, I would like to thank the members of the Mari Carmen Collado's lab in IATA, and also all of you for being virtually here. And I'll be happy to take any question or discuss with you at the end of the talk.
Thank you, Dr. Selma Royo, for an excellent talk. Yes, we are looking for the discussion at the end. And but now I would like to invite Dr. Collado for her talk. Dr. Collado is a research scientist at the Department of Biotechnology, Institute of Agrochemistry and Food Technology at the Spanish National Research